Hello, my name is Madeline Greff, and I'm going to be presenting on the cell cycle regulation. So the goals for this lesson will be to review the cell cycle, evaluate the driving forces of the cell cycle, investigate the checkpoints, review the regulation of CDKs, and examine the unregulated cell cycle. So the cell cycle review, what is the cell cycle? The cell cycle is the process in which the cellular components are doubled and then split into two daughter cells. The cell size control is critical for regulating nutrient distribution and organ size. If a cell becomes too large, the surface area of the cell is not large enough to be able to transport nutrients and to allow the cell to be functioning properly, so the cell needs to split. There are also in multicellular organisms, they have the need to control the amount of cells to ensure that tissues and organs are functioning correctly. An example of this is skin cells, which would always be dividing. But then you also have cells such as neurons that never do. So the cell cycle consists of two phases, interphase and mitosis. And then interphase is split into three sections. You have the G1 phase, which is the first gap, the S phase, which is synthesis, the G2 phase, which is the second gap. And then that leads into mitosis. So G1 phase is when the cell is performing the checks to assess the need and possibility of cell division. If it's not possible or there's not enough need for the cell to divide, the cell will then go into the G0 phase. It does need to divide, it will head into the S phase, which is where the DNA replication occurs. The G2 phase is then performing checks again to make sure that the cell can safely transition into mitosis. This is where the DNA is scrutinized to make sure that it looks correct. And then mitosis is the actual physical cell division of the cell. So interphase. Interphase is when the cell decides whether to divide. Enzymes and nutrients are synthesized that are needed for the division. The duration is variable depending on the nutrients available and the type of cell being divided. This is also when cells are going to be making the most proteins. So there's subphases in this G1 phase, the competence, the entry, progression, and the assembly. Competence is the absorption of the nutrients from the outside. Entry is when they enter the cell. Progression is the cell growth, and assembly is when all the parts come together to complete G1. So S phase, like I said before, is when DNA replication takes place. This is when the DNA separates with the DNA helicase. The DNA polymerase will read the template. A Y shape is formed, which is referred to as the replication fork. And replication does occur from the 5' to the 3' end. And then G2 phase is where new proteins are synthesized and the cell doubles in size before heading into mitosis. Okay, so mitosis. Mitosis is then split into 4 phases as well. This first phase that you see here, this is interphase. This is where the DNA has replicated. Then you head into prophase. This is the thickening and coiling of the chromosomes. The nucleolus disappears. The nuclear membrane disintegrates and the spindles start to form. In metaphase, the chromosomes will line up at the midline of the cell. Anaphase, each pair will start to separate into identical chromosomes pulled to opposite ends of the cell. And then telophase is when the chromosomes decondense, the spindles break down, the nuclear membrane starts to reform, and the cytoplasm does divide. And then cytokinesis is when you get two separate daughter cells. All right, so what are these driving forces of the cell cycle? There are really two driving forces. One would be the cyclins, which are proteins, and one is the cyclin-dependent kinases, also known as CDKs. These are serine, threonine protein kinases. Over on the left, you see the cyclin and the CDKs combine, they get phosphorylated, and then they bind to their target protein, which activates them. After the activation and their use, the cyclin is then destroyed, which makes the complex inactive. Over the cell cycle, the cyclins have different concentrations depending on which phase you're in. You'll see cyclin D is kind of throughout the entire cell cycle. Cyclin E peaks between G1 phase and S phase. Cyclin A really starts to build and then peaks into G2 phase, and cyclin B peaks into mitosis. This is important because each of these cyclins are responsible for stimulating different sections of the cell cycle, so you don't want them at all times. You kind of want them at specific times to make sure that everything is firing correctly. So CDK and the cyclins. So CDK7 cyclin H complex is also known as CAC. It presents in all the phases. It is responsible for the activation of CDK1 to enhance the cyclin binding. There are three interphase CDKs. These are CDK2, CDK4, and CDK6. And then you have a mitotic CDK, which is CDK1, a cell division control protein, also known as CDC2. There are 10 cyclins that are broken up into 4 classes: the A cyclin, B cyclin, D cyclin, and E cyclin. So throughout the cell phase, you have these different checkpoints. These checkpoints are making sure that the cell cycle is progressing correctly. You need certain signals to continue past these checkpoints. The cell will look at external and internal cues and decide whether or not to move forward. G1 and G2, the length of G1 and G2 will depend on how large the size of the cell is planning to be. And then you also have the meiotic spindle, where segregation of the sister chromatids in the anaphase. The spindle is composed of microtubules and motor proteins. The anaphase promoting complex promotes separation of these sister chromatids and targets the meiotics. Cyclins, they abolish the CDK activity and allow separase to be released and cleave cohesion complexes. The difference with the meiotic spindle is here the CDK activity is maintained to halt the cell cycle transition, whereas in interphase inactivity of the CDK is what halts the cell cycle. Yeah. So G1 checkpoint is also known as the restriction point. This is really the checkpoint where the cell decides whether it's going to start replication or not. A few things that the cell is looking for at this point: it's looking for cell size. Is the cell large enough to decide to divide? Nutrients, are there enough energy reserves to be able to divide? Molecular signals, is it receiving positive cues? These are things such as growth factor. Molecular signals, DNA integrity, is the DNA damaged at all? And then mechanical cues. So the cells exit the cell cycle and enter the reversible or permanent G1 phase, which is regulated by cyclin C, CDK3. Various extracellular signals, such as the mitogenic signal, lead to the synthesis of cyclin D and stimulate CDK4/6, which results in promoting entry back into the cell cycle. This active CDK4/6 complex initiates the phosphorylation of RB protein, thereby unleashing the E2F transcription factors. These result in the expression of cyclin E, cyclin A, and cyclin B, and many other genes that are going to be required for S phase progression. Cyclin E subsequently activates the CDK2 and contributes to the further phosphorylating RB, which progresses into S phase and initiates the DNA synthesis. Okay, so like I said, G1 phase is really cell growth. The cells are going to exit the cell cycle. This is kind of what I talked on in the last slide as well, kind of have it all laid out. That cyclin D is what activates the CDK4 and CDK6, phosphorylating the RB protein, which activates E2F, which completes the cascade down. All right, so the G1 checkpoint. So this is where, if there is any kind of DNA damage, the cell does not want to continue. During normal cell cycle progression, the transcription factor P53 remains unphosphorylated and is targeted to the proteasome for ubiquitin-mediated degradation by the MDM2 binding. As a result, P53 is unable to efficiently activate its target genes. Active cyclin E, CDK2, and cyclin A, CDK2 complexes accumulate in the cell, allowing the cell to initiate and complete this DNA replication. Following DNA damage, however, P53 is phosphorylated at serine 15 and serine 20 by the ATM and CHK2. This results in the displacement of the MDM2 and mediates both the stabilization and accumulation of the P53. What this means is the P53 is then free to activate transcription of its target genes. This includes P21, which accumulates in the cell, binding both cyclin E and CDK2, and the cyclin A, CDK2. This inhibits them, which accumulates in the cell arrest. P53, G1 phase halts replication so that DNA repairs can also be made. DNA repair enzymes are also activated by the P53. If the DNA cannot be repaired, it can also direct the cell fate to apoptosis, which is cell death, or senescence, which prohibits the cell from ever dividing again. Okay, so S phase. So everything looks good. The DNA is not damaged. The cell is getting all the signals that it's supposed to be getting. It will proceed into the S phase. S phase is the portion of the cell cycle where DNA replication takes place. Helicase will separate the double-stranded DNA for replication. It creates a replication fork, which you can see right here. Primase places RNA primers that are the starting location for the elongation. The enzyme DNA polymerase is what controls elongation. After elongation is complete, 2 new double helices have replaced the original helice. And enzymes called nucleases proofread the new double helix structure and will remove mispaired bases. The DNA polymerases then fill in the gaps created by the excised bases. Cyclin A and cyclin CDK2 activate during the late stages of DNA replication in S phase. The cyclin A displaces the cyclin E from the CDK2 cyclin E complex. The G2 checkpoint. So this is the DNA damage response. This is where the cell is going to be checking for DNA damage and DNA replication completeness from the S phase. They're looking for breaks in DNA, which would activate the ATM and the ATR proteins. The ATM-ATR signaling pathway is activated, which would lead to the phosphorylation and activation of the checkpoint kinases, CHK1 and CHK2, and to the subsequent phosphorylation of the protein CDC25. Phosphorylated CDC25 is then held in the cytoplasm, which prevents it from activating the cyclin B CDK1 complex. By doing this, it's stopping that complex from signaling the cell to go from G2 to mitosis, halting replication. In addition, the P53 also induces the transactivation of the P21, which also is an inhibitor for the cyclin B. Here, it's also monitoring the DNA replication. Replication origin firing is controlled by the CDT1 and the CDC6 proteins. When these are phosphorylated, replication is initiated, but then these proteins do go through degradation so that they cannot replicate from this origin again. The next stage after this would be mitosis. Cyclin A and CDK1 complex will transition the cell from G2 into mitosis. When the nuclear envelope degrades, so then does the A cyclins. Cyclin B and the CDK1 are then the driving forces of mitosis. And then the last checkpoint we have is the spindle checkpoint, or also known as the M checkpoint. This is where the cell is checking for chromosome attachment to the spindle at the metaphase plate. It's looking for chromosomes that would be in the wrong place in the cytoplasm. So it doesn't actually monitor the metaphase plate. If it finds that the chromosomes are in the wrong place, if they're not where they should be, it will be paused to give time for the chromosomes to line up correctly. All right, so the maturation promoting factor MPF is CDK1 bound to B cyclin. The B cyclin builds as the cell approaches this M phase, binding to the CDK1 that is present. The MPF will add phosphate tags to several proteins that will begin M phase, which is the membrane breakdown. MPF building up also activates the APCC, which will actually inhibit MPF then. So APCC is a protein complex that will then destroy the B cyclin. It works by adding a small protein called ubiquitin. It sends targets to the proteasomes for destruction. It sends B cyclin to the proteasome, which allows the cell to exit mitosis and the daughter cells to enter the G1 phase for continuation of the cell cycle. It is signaled during metaphase to set off a reaction for cohesion breakdown. This is the protein that is holding those sister chromatids together. It adds ubiquitin to the protein securin, which normally inactivates separase. Once the separase is no longer being inactivated, it chops up the adhesion that holds the sister chromatids together. You can actually see this on this slide. So here is where the spindle checkpoint is on. It's not allowing it past. So you'll see the APC is inactive. The securin is inactivating the separase, so the cohesion stays intact. On the right here, you'll see the spindle checkpoint is off. The APC is active, which means it's activating separase by inhibiting securin. With separase activated, cohesion is cleaved and the sister chromatids do go through separation. All right, so the regulation of the CDKs. So we did start to talk about this in the past slides. The production of the cyclins and the CDKs are regulated by the presence of the growth factors and the cytokines. Cyclin production, the concentration changes throughout the cell cycle depending on what phase the cell is in. There are always CDKs present in the cell. The cyclin concentrations are actually what are activating the complexes. The CDKs are also controlled by the degradation of the cyclin subunits. This was also discussed in the past slides. This example of what this was when in S phase, the cyclin A displaced the cyclin E, which then would lead to the cyclin E degradation. Once these cyclins are degraded, they're not able to continue firing off the phase that they have been activating, which allows for the cell to just continue through the cell cycle in a nice fluid motion. All right, so inhibitory proteins. The CDKs are also negatively regulated by the inhibitory proteins known as CKI or tyrosine phosphorylation, which blocks the phosphate transfer to the substrates. So CDK inhibitors or CKI, CDK binding or CDK cyclin complex binding is by two main families. One is the INK4 family. These are proteins P15, P16, P18, and P19. They work by inactivating the G1 CDK cyclin complexes, the CDK4 and the CDK6. This prevents the association with these CDKs with the cyclin D. The CYP-KIP family are made up of proteins 21, P21, P27, P57. These work by inactivating the CDK2 cyclin complexes, so it inhibits from binding with the cyclin D, E, and A. And the last regulation of the CDK is the phosphorylation and dephosphorylation. So full activation by the CDK7 cyclin H complex, also known as CAC. The CDK1 requires the phosphorylation of threonine 161. CDK4 requires phosphorylation of threonine 172. And CDK2 requires the phosphorylation of threonine 160. So these do need to phosphorylate to be able to be activated. And then you have the WE1 and the MYT1 kinase phosphorylate the CDK1 inactivating this kinase. So it's not just about phosphorylating or dephosphorylating. Both of them kind of work to inactivate and activate. And then the dephosphorylation of the CDK1 by the CDC25 reactivates it. So unregulated cell cycle. So the severity of the cycle cell defect could be something that could cause dysfunctions as severe as leading to cell death or cancer. Tumors and cancers are the result of uncontrolled cell division. The direct cause is almost always a genetic defect in one or more of the proteins that regulate the cell division. In some cases, a defective gene is inherited from one parent. In other cases, it occurs when the mutation is from a toxic compound from the environment or from radiation. Radiation that interacts with the DNA to damage it and introduce a mutation. In a lot of cases, it's going to be both inherited and environmental contributions. Excuse me. And it's not just one mutation that's causing this. So an example of some of this, some of these unregulated cell cycles, a p53 loss. This is an example of a tumor suppressor gene mutation. This p53 loss is the most common genetic defect in cancer, and several decisions are affected. So the lack of the CDK control by the p21 means a loss in the G1 checkpoint. The cells will continue dividing. p53 also allows for DNA damages to be repaired by halting and activating the repair enzymes. So without this step, not only do you have the cells continuing through the cycles without regulation, but it's not pausing to fix mutations, which allows the mutations to accumulate more quickly. And then the RAS mutations, this is an example of an oncogene, because this is a mutation in the signaling proteins. So mutant forms of the G protein RAS are common in tumor cells. The RAS oncogene encodes a protein with normal GTP binding, but it does not have any GTPase activity. So it's always in its activated form, regardless of the signals that it's receiving from around the cell. The results can be unregulated growth. Mutations with these are associated with lung and colon carcinomas and more than 90% of pancreatic carcinomas. So in conclusion, the cell cycle is a tightly regulated cascade of events that is signaled by external and internal forces. This regulation allows for the successful and complete replication of the DNA. The CDKs and the cyclin complexes are the driving forces that catalyze the reactions and stages necessary for replication. And the unregulation of these steps can lead to detrimental effects in the form of tumors and cancers. But understanding how the cell cycle is regulated and how the unregulation is caused can lead to treatments that are able to target these damaged or absent controls. Here's a list of my references, and thank you.